All right, welcome back to our second video in our genetics module. With this one video, we're going to go through test crosses. We're going to talk about the rule of multiplication, and then we're also going to talk about some genetic disorders. And then in a final video, we'll go over the incomplete dominance and some other unique situations that can happen during genetic inheritance. All right, so the last thing we had kind of finished with in the first video was talking about the dihybrid cross, and we went over that Labrador Retriever example. And what we want to do now is kind of look at what's called a test cross. And so a test cross is where you have a mating between an individual that has a dominant phenotype, but we don't necessarily know what its genotype is, and a homozygous recessive individual. So continuing with our example from the lab example, we know that black coat color is a dominant genotype or a dominant phenotype, and we know that brown coat color is recessive, and so that means that the dog with the brown coat color can only have one genotype. It can have homozygous recessive or lowercase b. But now the black phenotype could have two potential genotypes. It could have homozygous dominant, which is both uppercase B's, or heterozygous, which is an uppercase B and a lowercase b. So now we have to determine which of the genotypes are through a test cross. And so what that means is, if we do a test cross and all of the offspring are black individuals, then that means that the genotype for the black parent was homozygous dominant, because that means they only give a black allele, and so all of the offspring will be black. Now, if the genotype for the black parent is heterozygous, then half of the offspring will be black and half of the offspring will be brown. That is because the parent will be giving a dominant allele and a recessive allele. And so we can do the test cross to determine exactly what the genotype of the parent is. And so now this is useful because it allows us to create and develop pedigrees where we look at how alleles may have been passed through family trees and family history, which we'll talk about in a few minutes. What Mendel also did with his research is he had, this pretty good background in math, which allowed him to understand the patterns of inheritance a lot better, that, allowed him to have a greater understanding of the patterns of inheritance that he was observing. So he understood that these genetic crosses will obey the rules of probability, and these are the same rules of probability that you would learn if you took a basic statistics class. So like tossing a coin or rolling dice or probability of drawing certain cards, and that you can take those probabilities and apply it to the rule of multiplication, and which basically says that the probability of a compound event is the product of their separate probabilities. Okay, so basically, if you have, say, a male who's heterozygous and a female who's heterozygous, that the probability of the capital B being passed on is 50% or 1/2, because you can either pass on the capital or the lowercase in both situations. So that means the outcome in which you get an uppercase B and an uppercase B is 1/2 times 1/2. So that'll give you a fourth. So you have a 25% chance of getting both uppercase B's and not any other combination. And so that's the same thing as if you were flipping a coin. Probability of obtaining two heads is 50% times 50%, or 1/2 times 1/2, which is 1/4. And so that chance of segregation and fertilization is kind of follows that same idea and the same concept of the rule of multiplication with these probabilities of, say, flipping a coin. And so by understanding that, you can then look at these monohybrid crosses and you can determine the probability of any of the scenarios that we could potentially ask you that could come up with, say, this combination of heterozygous versus heterozygous cross of parents with this or monohybrid cross. All right, and so we can take these probabilities, we can take the understanding of our test crosses and the understanding of how alleles are passed from parent to offspring, and we can analyze the past family history of organisms. And this mainly comes from the fact that it's really not ethical for geneticists who study people to control the mating of their research participants, right? So instead, what they'll do is they'll go in and they'll get as much information from the people in the family tree, and then they'll assemble what's called a pedigree. They'll assemble that information and develop a pedigree. And so we can use Mendel's laws that he developed in order to determine genotypes from a majority of the people in the pedigree. And we say a majority because sometimes the genotypes cannot be discerned based off of the information that we have available to us. Okay, so in this example with this pedigree, we are looking at the phenotype of having freckles versus not having freckles. So freckles is a dominant phenotype, and no freckles is actually recessive. Okay, so if we look at the parents, Aaron and Betty, they are heterozygous for freckles. Okay, so that means that they have freckles and they are expressing that dominant phenotypic trait. Their offspring, Fred, Gabe, Hal, and Evelyn, will have specific genotypes based off of their parents' phenotypes. Okay, and what we can do is we can look at these relationships and we can deduce what those genotypes are. Okay, so for example, if we did not know Gabe's phenotype, if we did not know Gabe's genotype, then we could determine that because we have his parents' information. So we know that Aaron and Betty have freckles. We know that freckles is a dominant trait. And if Gabe does not have freckles, then that means that his genotype has to be homozygous recessive because that's the only way no freckles can show up. And now if he's homozygous recessive, then that means his parents have to both be carriers of that recessive allele, so they have to both be heterozygous, okay? And so anytime you see the phenotype of the recessive trait, you know for a fact that they have that recessive genotype. And so you can use that information to determine what their genotypes are. But now the one that we can't determine is Evelyn and Lisa because we just don't have enough information. So we know that their parents are heterozygous, but Evelyn, because she has freckles, could either be homozygous dominant or heterozygous. The only way we would be able to find out is to basically track her information and see what the phenotype of her offspring would be. If her children did not have freckles, then she would be heterozygous. If all of her children have freckles, then it's most likely she's homozygous dominant. And so we would have to kind of follow her life to get that information to determine her genotypic or her genotype for that character. And so we can use that information to build pedigrees of family trees to see how these alleles are being passed. And one thing I want to point out is that just because we say a trait is dominant, it doesn't mean that it's the most common or the normal or average phenotype that you see in a population. The only thing that dominance means is that if you have this allele with another allele, that the dominance allele will be expressed over that recessive phenotype. Whenever we refer to traits that are seen most often in nature, we refer to those traits as wild type traits. So a wild type trait is a trait that you see most often, and it could be a dominant or a recessive trait. It just depends on which one is seen most often. And you'll see kind of how that plays out, especially whenever we talk about the genetic disorders that can occur and that can show up in human populations. All right, the next thing we want to mention is that a lot of the characters that we've been looking at has been solely driven by a single gene, and that really reduces the complexity of how those phenotypes are expressed. But now there are a bunch of human characters, for example, eye color, hair color, and height, that are really complex and a little bit more difficult to understand because these genes or these characters aren't really tied to a single gene. They are generally tied to multiple genes, and that means that the inheritance patterns are not as simple as the ones that were developed by Gregor Mendel. And so Mendel's laws can be applied to human traits that are determined by a single gene. So like when we looked at freckles and widow's peak, which we haven't looked at yet, but we will, which is basically the shape of your hairline, and some other human disorders that are dominant and recessive disorders. And so those are from simple inheritance patterns where, again, they're only controlled by one gene. But you can have inheritance patterns that are controlled by multiple genes, and those are way more complex and a little more difficult to understand and discern. And so again, a single gene trait or character is basically your hairline. The dominant trait is widow's peak, so you kind of have that V shape in your hairline. And then the recessive trait is a straight hairline, so where it kind of just goes straight across the forehead. And again, only controlled by one gene. Simple inheritance patterns, follows Mendel's laws and hypotheses. The next thing we want to discuss is disorders that can happen based off of the inheritance of alleles. Some of those are albinism, cystic fibrosis, sickle cell disease, Alzheimer, so one type of Alzheimer's disease, Huntington's disease, extreme cholesterol levels, which is hypercholesterolemia. And so all of these are really controlled by kind of single gene associations. Some of these are recessive. So like albinism, sickle cell disease, those are recessive disorders, but you also have dominant disorders. So like achondroplasia, which is dwarfism, or Huntington's disease, which is another dominant disorder. And what's really interesting about dominant disorders is you can get that disorder if you're homozygous dominant or heterozygous. The only way to not have that disorder is to be homozygous recessive, which is the complete opposite for those recessive disorders. That's the only way you can have the disorder, is to be homozygous recessive. So there's a little bit of interesting kind of interaction with dominant and recessive disorders. So the first thing we're going to look at is the recessive disorders. So most of the genetic mutations, the medical disorders that you're generally aware of, are in fact disorders that are caused by recessive genotypes. And what's interesting with recessive disorders is that individuals can be carriers of the disorder but not have any symptoms. So for example, if they're heterozygous. So for example, sickle cell. So sickle cell anemia only shows up when you have the homozygous recessive genotype. But you can be an individual that is heterozygous, so you have a dominant and a recessive allele, but you don't show any of the symptoms of sickle cell anemia, and your blood cells have the normal shape. But again, you can produce offspring if you have children with somebody else who's a carrier. And so you could have a potential, a 25% chance of your children having sickle cell disease at some point. Another example is cystic fibrosis, which is the most common lethal genetic disease in the United States. You have about 1 in 31 Americans, or about 10.5 million Americans are carriers, so they're heterozygous at the genotype for cystic fibrosis. And so we can take Mendel's laws that we learned in the first video for this module, and we can actually calculate the proportion of our offspring that will be affected by a marriage of individuals that are carriers. So for example, if we look at albinism, which is another recessive disorder, you have parents that are heterozygous, so they're carriers because they carry that recessive allele. They have normal pigmentation in their skin. And if they have children, 1 out of 4 children will have normal pigment and be dominant homozygous, so they will not be a carrier. 2 out of 4 children will have normal pigment, but they will be heterozygous. They will be a carrier. And 1 out of 4 of their children will be albino, so they will have the albinism trait, and they will be homozygous recessive. So the phenotypic ratio would be 3 to 1. So 3 would be normal, 1 would be will have albinism, and then the genotypic ratio would be 1 to 2 to 1. 1 is a normal non-carrier, 2 is heterozygous carriers, and 1 would have the homozygous recessive disorder. And so if we look at this question here, a man and a woman who are both carriers of cystic fibrosis have 3 children without cystic fibrosis. If the couple has a 4th child, what is the probability that that child will have that disorder? Okay, so based on the previous 3 children not having it, and what is the probability of the 4th child having that disorder? So take a minute to think about that. All right, so the probability is actually still 25% for that 4th child to have the disorder. Now, just because you have 3 children without it, it does not affect the percentage of the next child having it. Every time it's a 1 in 4 chance that your child will have it. Now, you could ask, what is the probability that out of 4 children, how many of them have cystic or have albinism? Then you would multiply 0.25 times 0.25 times 0.25 times 0.25, and that would give you the probability of at least 1 out of 4 having that that disorder, because you would multiply the 0.25 probability 4 times because you have a 25 chance every time, and that would tell you out of 4 tries what's the chance of having 1. But every try is always a 25% chance that one of your offspring will have albinism. All right, the next thing we're going to talk about is dominant disorders. So dominant disorders is the opposite of your recessive disorders. So instead of being caused by recessive alleles, they're caused by your dominant alleles. So for example, dwarfism, which is also known as achondroplasia. This is a disorder that can happen if you have a homozygous dominant genotype, so 2 uppercase A's, or if you have heterozygous genotype. Now, what's the difference with the dominant genotypes or dominant disorders is generally the offspring that are heterozygous are the only offspring that actually survive and are born. Usually, if the offspring are homozygous dominant, the embryo does not survive and the pregnancy is aborted. Because that dominant allele is expressing a protein that causes the disorder. And so if you have two copies of that allele, you're getting twice the expression. And so, that concentration of expression is typically deadly for the embryo. However, if you only have one allele, you're only getting 1 times the expression. And so that's not deadly to the embryo, but it does cause the disorder to happen and that disorder to be present. So what that means is that if you have a person with achondroplasia, they have a 50% chance of passing that condition on to any one of their children because it is a dominant disorder. And so here we have an example. So if you have a person that's achondroplasia, then that means they're probably heterozygous, right? Because the homozygous dominant typically doesn't even make it through embryonic development. And then if they marry a normal person, then that means they're homozygous recessive. So if you do the cross, right, you have your homozygous recessive plus your heterozygous. That means that 2 out of 4 of your offspring will have the dominant allele. They'll be heterozygous. So 50% of your offspring are likely to have the achondroplasia disorder. Okay. And whenever we see dominant alleles causing these disorders, they're much more common than the lethal recessive alleles. One example of another dominant disorder is Huntington's disease, which is where your nervous system basically degenerates. And this typically doesn't happen until you reach an older age in your life. And what that means then is that this disease is not preventing an organism or a person from reproducing. And because it happens after the reproductive stage of that person's life, then that person is able to reproduce, pass on its alleles, and then it gets affected by the disease. So because it's a later stage in terms of life disease, it allows for those alleles to be passed on to the next generation. And so it tends to stay in the population because of that. Whereas if it was a disorder that, killed the offspring before reproductive maturity, then the disorder would probably be at a much lower percentage in the population than, say, something like Huntington's disease. And so any child born to a parent with Huntington disease is also going to have that 50% chance of having it because it is the parent that has Huntington's disease is going to be heterozygous for that disease itself. Okay. And so that's it for the second video. The last video we're going to have in the module is going to be looking at variations in Mendel's laws. So variations from these simple single gene mechanisms where we have complete dominance, so dominant versus recessive. We're going to look at things like codominance and incomplete dominance and look at what that looks like in terms of genetic inheritance and the expression of phenotypes in offspring.